We are going to continue to record this session. None of us is mic'd, but we've got the microphone array, and that should pick up okay. So I just ask people to speak up, please, and that should be recorded okay. And you may notice that the cameras do swivel. So I hope that everyone in here is prepared to be, I should have given you all pieces of paper to sign to say you're prepared to be videoed. Anyway, good. So I don't need to introduce anybody on our panel except for Jane Rogers. Jane is from the Genome Analysis Centre. It's a BBSRC Institute in Norwich. Jane, do you want to just give us a minute about things that interest you, please, before we start?
The Genome Analysis Centre was launched last year as a new centre to primarily direct ourselves towards non-medical genomics, and to develop applications of genomics in areas such as plants, animal health, food security. As I say, the non-biomedical areas of bioscience in which there is a great deal of research in the UK. We use next generation sequencing platforms, a variety of them, and are facing issues ranging from generating genome sequences for organisms that don't have highly curated genomes in databases at the moment, through to working with transcriptomes and metagenomic samples as well. So sequencing sort of mixtures of organisms in different populations.
Great. So I'm hoping that this will degenerate into a really interactive session. And I mean, I have some questions which naively I could ask, but much better that things start coming up, obviously, from the floor and from the panel. And I just hope that everyone will feel that they can participate in this. So I think the best thing I can do is just throw it open and look for anyone who wants to start the ball rolling. Otherwise, I will. Please.
So a couple of speakers spoke about the idea that maybe in 10 years' time there'll be kind of routine population-wide genome sequencing. I was just wondering if before that happens there will be subsets of the population where it becomes routine. I'm particularly thinking about people enlisted in clinical trials, and whether Matthew thinks that will become routine to say whole genome sequence, say 1,000 people that go through phase 1, phase 2 clinical trials. Mm-hmm. I mean, even at $10,000 per genome, the cost of that relative to the total cost of producing a drug is quite small, it seems. So if there's value there, yeah.
Okay. Well, I mean, at $10,000 a person, I guess the cost of that is not, it's not relative to the cost of developing the drug. It's still a significant impact. I mean, we are very cost conscious in the pharmaceutical industry. The question is, what's the potential value of having those data? So, I would see in the next, if the data from those could even include, say, HLA and the cytochrome P450s, let's say, where it probably is challenged to do so now, I think we would still have a hard time arguing for generating those data, because not every clinical trial generates a hypothesis that we feel should be addressed by genetics. So probably the majority of trials do not. Now, if we reached a point where virtually everyone within a medical system has whole genome genotype data available through resequencing, then we might be looking at a situation where we would want to incorporate that's already existing for that patient. And that could perhaps open up the doors for more exploratory types of analyses. But even today, with $300 to $500 genome-wide genotyping platforms, we're still not even considering the option of genotyping everyone in a clinical trial. There's just not enough value per trial, per patient for that kind of data.
Yeah, certainly come in and endorse that, I think. And clearly I'm hearing the message that $10,000 is too much. We've got to work hard to get it down. But clearly I think to pick up on an element of what Matthew said is in a clinical trial you're looking at a very specific phenotypic piece of information on indeed a relatively small number of people. But if you can somehow capture that much richer phenotype information from a more general population sample, nothing is perfect, but the electronic record will capture everything that somebody has at least found relevant to record. That's a whole range of data, and therefore potentially increases the power to detect these new associations and attach functional value to variants. So I think these more general populations are really important. And I do think, in the meantime, you asked about intermediate groups. I mean, the biobanks clearly are trying to embrace this as well. I mentioned at the time, but I reiterate it now. They're trying to accumulate more phenotype data per individual, as well as making those individuals fully consented and available for such studies, which hopefully will be the forerunner, perhaps, to the individual medical record being available for research for everybody.
Any other comments or questions from anyone? Okay, onwards. Please, yeah.
So we saw Nigel talk about the utility of genotyping trios, so the parents and the offspring, for kind of highly penetrant, rare disorders. Is there utility of that in the general? So the other speakers talked really about kind of population-based approaches. Do people see utility in being able to sequence on type pedigrees on small family units?
I could take that from a straightforward technical viewpoint. Absolutely, yes. I mean, a trio gives a very immediate way of looking at observations that should occur more than once in the trio, and that gives, as you will know, but the algorithm development is a long way to go. To have that kind of genome-wide information is incredibly valuable technically for this kind of study.
But that sort of to make that kind of cost-effective, because there's a lot of duplicate information, but you sort of lower the coverage, you know, to on each individual, I guess, because of the.
Actually, for my particular view, I wouldn't, because we're looking at an algorithm that is being tuned to what you'd call a comprehensive depth. So if you say you set up the experiment to sequence an individual to a high level of completeness and accuracy, you choose a level of, say, 30x or 40x, which is going to cover 99% of the genome or 97% of the genome effectively. You then, if you're using another set of data or several data sets to calibrate those algorithms, you don't want to compromise the actual data you're using. So it's more a question of analyzing each one separately and then looking at the expected concordance and discordance to look at how.
Yes, so it's a question of what you would use a trio information for. I mean, you can use it as a method for sort of an incredibly useful data set for learning about variant calling, because you can say anything that sort of breaks the transmission rules is a false positive, which isn't far wrong. Or, you know, there's a version of it where you could actually do disease genetics. You can look for de novo mutations, or you can do TDT type things. Do you, Chris, what would you want trio data for?
I was just interested in the fact that we had one of the speakers talked about, you know, the utility of trios for a specific kind of application. I just wondered what the panel's view was on kind of wider applicability of that approach.
It does. I mean, a totally different question. I mean, did you want to pick up on this?
Well, I'm just going to say that it's a specific use of the trio data in this cohort of patients, because one of the clinical criteria of a variant being pathogenic is that it's de novo, and so that's the key data that we're looking for. But you know, as the others have said, there's other ways you can use that trio data to really maximize the information you can get out of the study.
It's triggered in my own mind because I bounce around. Just for Jane, is there any parallel in, for example, in animal genetics for breeding in the kind of question that we're discussing now? And we know that from the radio this morning that clones are extremely valuable beasts. They're worth breeding even if you pour the milk away, allegedly, right? So is that based simply on, you know, the traditional breeding of a quality animal, let's say, or is genomics now going into genetic sequencing now going into animal breeding?
I think to date most of it's been based on traditional methods, but they're now starting to use genomic methods. The draft genome sequences of the cow have been used to generate genotyping chips, which are used quite widely now in order to try and develop the pedigrees. But now they want to go down to the whole genome sequencing, and to actually then you just characterise the
More several breeds of cow being sequenced right now.
I'll just comment on pharmacogenetics. I mean, for the most part, family data is of limited availability for pharmacogenetics. There are only a few studies you could envision where you could actually collect trio or family data. Certainly, drug development in clinical trials, there's not access to family data. But for some drugs, such as commonly used drugs like statins, there are opportunities for family studies, but they're limited.
They
Would
know
you
like
want
the
family
heritability.
data? I don't think we've sort of really pinned this down because I mean, it's clear if you're looking for de novo mutations, then that's obviously quite useful. But in general, you're not particularly looking for de novo mutations in pharmacogenetics.
Not generally.
And you've got the obvious kind of wastefulness that once you know the two parents, you've got the entire child's genome. It's only the recombination points and the de novo mutations, which is a relatively small amount of information. So it seems relatively wasteful to sequence trios, except in those special situations. So I would have thought in your application you probably wouldn't particularly want them.
Well, I just was commenting, we rarely use trios in
You really
pharmacogenetics.
have them anyway, so it doesn't matter.
Presumably they would be useful in terms of estimating like heritability of adverse reactions.
Yeah, but the reality is you rarely have the same drug given to enough family members to be able to estimate that also have the adverse event. So,
Do you not care about it for phasing? The phasing of rare things, for looking at haplotype effects, that kind of stuff?
It's certainly not impossible that the haplotype context of a variant could influence, say, its expression effects. You might have sort of cis interactions, but so I mean there is some value, but it might be rare. I think there's an argument that says if we're sequencing enough people, then you won't have immediate family relatives in your set, but you'll have people who are like second cousins or something like that. So many of the things that you'd like families for example, estimating heritability or phasing, you can actually get by finding these individuals who are pretty close relatives out there. It's the approach that essentially Iceland and Decode have taken. So the only place in which that breaks down is for the truly private mutations, where it's me and only me that has that. Those are hard to do anything with.
I have a question. You're talking about trios and utility of trios, and I'm interested in disease, identifying disease variants. And let's say I have a large family with 8, 10 affected people, and that tend to behave as a single Mendelian disorder, although it is, let's say, Alzheimer's disease, which tend to be common, but there are some forms that are relatively rare and have early onset, so forth. If you have such families that are, let's say, 10, 20 families of that sort, would you sequence one person from each family and sequence the heck out of it, and then create a customized chip to look at those variants, or looking at the effective relative care type of approach where you take the distant relatives who are affected and sequence them in order to reduce the number of signal to noise ratio? I guess it depends on the cost issues. How do you go about that?
I could take a stab. It's difficult. It'll depend on case-by-case basis, but I think one of the interesting examples is if you are strictly adhering to a strong Mendelian pattern of inheritance. One of the great values, as with the early linkage studies, and as really shown recently by the Shinduri studies, which somebody quoted today, is having independent examples of mutations that affect the same gene, where you very rapidly... Was that your talk?
No, it wasn't me, but I'm agreeing with you.
Sorry, yes. Where if you look at one individual, you're going to have a whole lot of candidate mutations that lie in genes that could be obvious functional. If you then sequence another individual from the same family, you're going to get a lot of the same mutation profiles because of the relatedness. Take the same investment and put it in another individual with the same phenotype, with the hypothesis that the mutation is in the same gene, you are going to eliminate all but the common factors, which are really a much smaller subset shared between those two.
But that depends on how related, how close.
Sure. So the thing I wanted to bring out about the Shinduri studies is there were 2 papers from that group. The first, I think, was the Freeman-Sheldon syndrome, I think that's the one where they already knew the mutation. And they had 4 unrelated individuals and they homed in on the right gene. They then did another study on a disease they didn't know the causal mutation for in advance, and they actually had 2 relatives from the same family. I think, in fact, the power of the study was pretty good with the 2 SIBs and 2 or 3 others, but it illustrated this concern. I think they eliminated less in that comparison of the 2 SIBs than when they were able to take care of the unrelateds. So that's perhaps the nearest example I can think of to your question. The answer for me, therefore, would be, depending on the Mendelian pattern of inheritance and other information about the disease, I would go for the unrelateds first, and then look to back up inheritance patterns later on.
Well, I think you're right, though, that I guess in general, close relatives are going to have too much sharing. Because you're going to have someone more distantly related to narrow down what's causal.
Yes, I think the bottom line in that is, a linkage study with 1,000 microsatellites across the genome in a pedigree will get you as good a resolution as you can get, right? In that case.
So I mean, I would personally do it with the genome link study and then resequence the regions of my interest. But
the point still holds that you'll have a lot IBD anyway, and it's only by having independent information. The problem with going to another family is that it might be a different gene.
Please.
I want to pose the original question again of should I have my genome sequenced, but change it to when should I have my genome sequenced, and in particular what age should I be when I have my genome sequenced. A number of the panel have suggested it would be useful in a medical sense if the data were just there and we can go and interrogate it, whether it's on my iPhone or in a database. And equally it's been suggested that all newborns should be screened and it be stored. I can think of lots of good reasons to do that and lots of good reasons not to do it. I wondered what the panel's opinion.
Good and that.
As I think I tried to illustrate, it is a fascinating question. I love the idea of when rather than if, because I think it probably is becoming a reality in some form or another. We all do embrace in a very positive way. There's something valuable to get out of this, which was a message both from my experience and indeed from Gil at the end. There's lots more to get out of genome sequence. I think it is down to circumstance. If I was actually diagnosed with cancer tomorrow, I would say, please, sequence my cancer. I want to know anything you can do to help. And I certainly wouldn't be expecting a cure magically as a result of it, but I think people in certain circumstances really change the value of what they feel might help and ask people to go a great distance. If it was my newborn child, I'd be more nervous. I guess it's a little difficult because having a natural genetic interest, I'd probably be fascinated to do it. But I would at least have to, I would have to think very carefully about the ethical and moral situation of doing that to a minor. In fact, within the early days of establishing the whole genome sequencing service in Luminar, the one thing that our advisory board suggested we should do very early on is medically relevant samples, including minors, because we'd learn a lot from that. The one thing they said we should not do for a long time was well minors. There's a bit of a conflict there, because a well minor is somebody who hasn't got ill yet. And in fact, there was a lot of pushback on that, in fact, interest from physicians that we should sequence minors where there was family history of disease. So I think sooner rather than later. The challenge is is in the grey area of what you can and can't interpret. And what you perceive as the relative value, either to you, the consenting adult, or the consenting individual. And that needs an awful lot of education and guidance to bring it to a level of standard that you're happy with.
I think another way of looking at it is how much good you're doing against how much harm you might be doing. And I think if, for some of the children with developmental disorders, if we gave clinicians even the exomic data now, they wouldn't know how to interpret much of what we're finding. In hopefully the study that I described in a few years' time will help answer more of those unknowns, and so the good that you would do would outweigh the harm. And that's the way I sort of think about it.
That leads naturally onto a thought which came to me as an outsider, that in reality, I think a lot of the cost in the future is going to be in the expertise to understand the information, and possibly in an even greater specialization of clinicians who can do that kind of thing for the kind of diseases which you're beginning to understand now. Is that a correct statement or not?
Yeah, let me pick on that a little bit. It does carry on with the previous question as well.
Yeah.
It's the cost is important to recognise. For example, I was surprised myself, but in hearing from pathologists in Harvard last month, this man had put up a table of current cost of genetic testing, and if you add together BRCA1 and BRCA2 CF, one of them, which, okay, maybe that's an unlucky individual, but it's only 4 or 5 gene tests. It costs about the same as a human genome today. It costs between $5,000 and $10,000 to get those tests done. So therefore, you don't have to look for a huge amount of new value to actually have a straight cost benefit today and in accepted terms. Move to Nigel's answer from previously. As soon as you do additional research studies like that, you can add in other things that are of high certainty or high predictive value that you didn't know before. And this is where this table of risk variants comes up. As you expand the set of risk variants, the value in the genome becomes greater. You could start off today with that table of risk variants being just BRCA1, BRCA2, CF, and that's already $5,000 worth. Now add in the results of Nigel's study and a few others and quite a lot of other things you already know about, the drug metabolizing enzymes, for example. You've already got quite a valuable table. It's a minimal amount of the human genome, but it's already proving cost comparable and eventually getting more beneficial. So that's kind of arguing the cost of saying sooner rather than later.
Just a point of, I have a question, some information, which is, if I sequence my sister's genome and report on BRCA, is that I don't have to pay anything in addition to the sequence? Is that true? Or would the fact that it's a sort of a protected test, the fact that can I just report it?
If you sequence it, I'm not quite sure.
Sorry, if I
Myriad
sequence
Genetics holds the patents on BRCA1 and BRCA2 genes, and they've been in several settings supported in that patent. So can you draw medical inferences from whole genome sequence in BRCA1 and BRCA2?
It's a patent question, right? There are two ways of answering the question, I think. One was the question, was there anything else to pay? And the cost of reimbursement covers other costs like consultation and downstream further testing or treatments or mammograms or whatever for BRCA1 mutations. In the question of patents, and I don't know the certainty here, but the thesis has been, or the patent has been contested at least in part, on the basis that if you are sequencing a whole genome, you are not targeting a breast cancer test, and you are able to report out on the information because it is not contesting the patent, because Myriad have claimed specific testing for mutations in BRCA1 and BRCA2 in connection with breast cancer. Whereas if you're analysing a whole genome, you are not specifically making a test that targets that.
That's
Sounds to
probably
me a little semantic, I must
Is
say.
there
but it's-
any lawyers in the audience?
well. It seems to me that having a major player in Illumina standing up and saying, well, you can do the equivalent of these 5 commercial tests, is adding a bit of a legal problem down the line. You're not worried that someone's going to come up and say, look, they actually are targeting these things. Presumably you want that very consideration is something you don't want to think about too much or push too much.
Oh, we'd like to think about it, actually. We'd like to make sure we get it right. But there's a difference between saying we can do those tests now and actually saying the only reason to sequence the human genome is to do those 5 tests, which is exactly the tension that you're voicing. And clearly we didn't sequence the genome to do BRCA1, but clearly we have to prove or find in the court of law that that's not the case, or indeed the patents may not be upheld.
Do you think this will happen? Do you think that Illumina will be in court sometime in the next 10 years over this?
Only with the other companies.
Well,
They're doing the same thing. So no.
I mean, the other side of having the whole genome is the whole area of incidental findings, that you're going to find things about people that they are not expecting and may not want to know. And not only about the person's genome that you've sequenced, but their family. BRCA1, for example, you could find in a 2-year-old girl a deletion of BRCA1. It's not really appropriate for that child at that age to have any sort of discussion of intervention. But the fact that that child has that deletion means that all other female members of that family may be at higher risk of familial breast cancer. They have not been counselled or given consent to have that sort of information available. And so what you do about incidental findings is very, very difficult. And what we're doing is we are getting consent that we will not report any incidental findings. So we're just sort of putting our head in the sand. These sort of things, late onset neurodegenerative disorders, whole range of things that people may or may not want to know about, but that you will inadvertently find.
So when you say you don't report the incidental findings, do you record them somewhere? Are they available to that individual's clinician, or you just basically write it off and pretend that you just didn't see it?
The fine details of how we do this are still being argued. I think, we will not look, but we're going to make the data available, so it will be there. But by making the data available, we have to have the protection that those individuals no one will seek to actually identify those individuals. So you have to put protections in place. But certainly the view at the moment is that incidental findings not related to the diagnosis of the particular child that we're trying to diagnose are not reported.
But then when the data will be published, the sequence data, presumably they'll be deposited in a repository.
Yep.
And presumably the phenotype information as well.
Yep.
So, I mean, what's to stop me taking that data, reanalyzing it, saying, oh, look, this young girl here has this deletion of BRCA2.
So what you would be able to say would be that X children from a population of 10,000 had deletion of BRCA. What you would not be able to say is this child from this clinician from this area has a deletion of BRCA. So it's the difference between identifiable linked anonymous data and anonymized data, which you could, I mean, if you wanted to, you could find that child, but the data access agreement will be signed where you state that you will not even attempt to do that. So that's the sort of way that these sort of issues are being dealt with or attempt to be dealt with at the moment.
But at some point, whole genome sequencing, nothing becomes incidental, unless you're doing it for the very extreme purpose of a very small number of potential genetic variants. Then you would consider all of it relevant to some extent. I would argue that there are probably very few genetic variants, let alone genes, for which we can properly interpret the genetic risks. Even BRCA1 and BRCA2, it's been shown that most of what we know about the risks associated with those genes are derived from families that have familial breast cancer. We really don't know, if you take 20,000 people from the population at large, if 30 of them have BRCA1, BRCA2 deletions, what's the probability that they would develop breast cancer given they carry those deletions? Because for the most part, the data we are familiar with is conditioned on a family history of breast cancer. So it becomes very difficult, and that's true, I think, for just about every gene and genetic disorder that we're talking about. It becomes very difficult then for us to put absolute risks on anything that we might derive from whole genome sequence, which is why in my presentation I'm starting to get some sense that if we really are going to get maximal use of this, it's going to be an interplay between discovery and prediction as we start to generate these data on a large scale.
It's a great example because it illustrates, from Nigel's comment, if you have a 2-year-old with a breast cancer mutation, you don't go and do something immediately preventative that is irreversible, although there have been cases of that in the past anyway. But what you do is you want to collect the information from as many individuals as possible to try to ascertain better what the true predictive value is. It's quite true. We've not looked at any large number of even the simple Mendelian disorders in the widespread population. Does a mutation in factor 9 really give you haemophilia B every time?
Cystic fibrosis. Cystic fibrosis.
So going on with that, I think a number of the speakers mentioned that it was really difficult to get phenotype information. So is the problem there that it's just not in a database that's accessible, or is the problem that people aren't willing to sign it over at the same time? I mean, because to know whether these genetic mutations are actually involved with a particular phenotype, you're going to have all of that information. So do you think that the people, A, are going to be willing to do that or give that information, and B, is it actually ever going to happen?
So it will happen prospectively for a study that wants to collect that data. What's difficult is that a lot of the cohorts that we have now, that data either hasn't been collected or it hasn't been collected accurately enough.
Any other questions? Yes, please.
As concerned the 1,000 genome project, how did you go around? What are the criteria you used to select the 1,000 individuals?
It's based on a combination of sort of scientific and logistical and ethical sort of principles. The idea, so it's quite similar to the HapMap project, if you've heard about that. So the idea is to sample broadly across centers of major medical genetics interest, but not to try and represent sort of the whole breadth of human variability. So that's why there's sort of targeted collection in a few areas rather than a sort of a very widespread. So that's one principle. The other was to collect a decent number from any one location. So typically 60 to 90 individuals as a minimum. The most important thing is that all these individuals consented for complete data, genetic data release, and they're consented for cell lines, immortalized cell lines, to be available in perpetuity through Coriell Institute in the US. So those are quite strong things that these people are signing up to, and for that reason it was decided not to try and attach phenotype information. Some of these collections are actually quite old, so that would have been quite hard, but going forward we're not attaching phenotype information. And part of the reason is, we're not actually talking about a huge number of samples. If you want to really understand the genetics of a particular, say, common disease, you need to be studying tens of thousands of people, really. And we're talking about 2,500 people from across the world. It's not enough information, really, so it wouldn't be very useful.
Yeah, my question, I was asking that because, like, in certain African countries, I mean, there are some ethnic groups which are susceptible to certain particular type of disease. Like if you go to Nigeria, there are certain regions where some people, they don't even pick the malaria and all the like.
So we very deliberately stayed away from some of these classic founder populations, or Ashkenazi Jews, for example, where there are high frequencies of certain diseases, for the reason that we want the data to be as much information to as many possible people. So it's not very focused.
Okay, yeah, David, and then, yeah.
I was just changing the subject completely, mainly for Nigel, but others might have a view as well about, do we have a story yet about, you took it for granted sort of throughout your talk that copy number variants were important, and yet you had this sort of striking result that, well, I don't know if you took it for granted, but it was a feature at various points, and whereas, you had this contrasting result that the CCC found common copy number variants to have virtually no association whatsoever. And is it? I mean, we've got to put a piece together a little bit of the story for this, but it still seems a little bit surprising. I don't.
So I think this is a case where size matters. So a lot of the copy number variants that are important for children with developmental disorders are large. So they're large deletions, large duplications. They involve multiple genes, and we assume that in these regions there are copy number sensitive genes. The vast majority of the copy number variation in the normal genome, and Gil will have more details than me, is at much smaller size. And also, the patients that I'm looking at have been ascertained because they have a very severe penetrant phenotype. And so there's a causative deletion there. Obviously, a severe penetrative phenotype wouldn't be common in the healthy population.
And so a moderately rare copy number variant is going to be important, do you think?
Well,
that,
you know, there's this overlappy sort of region where,
So where does it come in? We don't know.
well, I don't know if
There are obviously well-established and important structural variations associated with autism, intellectual disability, and so I don't have a huge amount of information, sort of intelligence on this, but the big things are clearly under strong selection. You look at megabases, that's going to be important. The small things that are segregating at reasonable frequencies, is your belief about the deleterious nature of those is going to be somewhat limited. And something like CCC was only assaying a few thousand. You know, we in a SNP study, you assay maybe 1 million now, and you pick out a very few of these that are important.
And so you think the distribution of effect sizes is different for CNVs than for SNPs?
It interacts with length.
Yes, because of the length.
Interacts with length.
Doesn't exist.
Another question, yeah?
So I think this is very interesting. But one thing that's not clear to me is what is the added value of sequencing when you're detecting really minor variants with a small effect, or rare variants with a strong effect, but it's private, and that phenotypic expression varies by the ethnic groups and the other environmental factors that are contributing, that modulates phenotypic expression. So if you were to sequence somebody who's 25, how's that going to help that person and the clinician for the future medical care?
I'm going to ask about plants in a moment as well. Same question. What's the added value? Who's going to have a go? Added value.
There are a number of points in your question there, and I guess if I was to focus on the final one, if I heard it right, it was what advantage would somebody who's 25, what advantage would be sequencing over other methods of analysis for an individual?
Whatever other method, conventional, suppose even doing a cheaper GWAS, because you were talking about lookup table of risk variance. So if I have a set of high risk variance with a high penetrance, that would be a heck of a lot cheaper, number one. And there are other issues, because you were sort of talking about less of a common disease, but more a strong penetrant disease. But this discussion is about sequencing the whole genome in a average person on a day-to-day
Okay,
basis.
so let me answer a few points there. I guess one question to contrast is whether you are looking to take a genome or an individual and test it for specific hypotheses that you know, that you have tests to. So that could be quite as simple as 3 CYP loci or the BRCA1 gene again, and that's a very specific test. You could argue extend that model to a wider panel, and it's much cheaper than sequencing the whole genome. That's perfectly fair, and you get some well-calibrated predictions that you know from past experience. As an aside, you can't really say, can you do a GWAS on a person? This is heard a few times. If you're taking a 1 million SNP chip and testing it on a person, you're not doing a GWAS, but you are looking at the 1 million specific tests on that chip for specific predictive value. The real value in going to the whole human genome is partly where you're testing the individual for anything that isn't on that chip to start with. So as soon as there is one very important variant that is not on that chip, the chip is essentially out of date for the maximal information, and you can do better. Secondly, and going on from that, you are investing for the future. It's not a one-off test for one moment. You're actually investing in information that does largely not change for your lifetime. And so in doing it once, it's a very technologically and cost-effective way of ascertaining genetic information. The cost of an array with 1 million variants is only about a 40th of the cost of the entire genome, so 1 million to 3 billion or 6 billion. That's a huge value add itself for the genome sequence. But the challenge is only interpretation. So do it now, if it's accurate enough. Should I sequence my genome now, or should I sequence it in 2 years? Technically, it's very accurate now. It's more accurate than the gold standard of a sequencing-based test. But the ability to interpret it will increase in value as research carries on investigating more variants. So for the 25-year-old, yeah, I would argue do the genome. Especially given there is a reimbursement and a structure that can do it. So maybe now is too early for the 25-year-old, maybe in a few years' time when more of that infrastructure is in place.
Yeah.
Is there some sort of estimate of how much is the contribution of genetic diseases to environmental diseases? So how much are we going to save in terms of human health if we knew about the genetics of individuals rather than having them run 30 minutes a day or walk hard?
Or have a colonoscopy.
Is it 1%? Is it 20% or something like that?
We know.
Diseases would be genetically.
So I think the answer to that is that most diseases have a genetic component. The goal of disease genetics is not to identify the genes. The goal of disease genetics is to understand disease mechanism, and genes are a route into that. So the idea is that disease genetics identifies mechanisms that you can intervene in irrespective of that person's genetics. I mean, the individual's genetics might be important, but it gives you an understanding of a mechanism that you can exploit for anyone who has that disease.
So that's a good argument to do research, but to do population-wide testing, that's a different question.
Yes, they're different questions. You know, understanding mechanism and in predicting an individual's outcome are quite separate. But I think, as we've heard, you know, a genome is a big thing. You're at risk. You're potentially at risk for an awful lot of things. There will be some things that you personally are strongly at risk for. You might quite like to know those. And so sequencing your genome, maybe not now, but maybe in a couple of years, you will be able to say these are the things that you are specifically at risk for. And there, in some cases, with a bit of luck, there'll be an intervention that you can use.
But I think behind your question, it is a very fair question on a couple of levels. One is, how much do we as society invest in this kind of technological approach versus investing in and perhaps creating a healthier environment so that we have fewer of these common complex diseases? And I think the other question that we're going to have to ask is, how much unnecessary medical intervention would we create by the kind of information that's going to be returned from this type of genetic test? This type of testing could drive our healthcare costs through the roof if we are not careful and informed about how that information is used. If everybody goes on 15 different drugs because their genome sequence says they're at risk for these different diseases that could occur at any given time, and they go through a series of prophylactic therapies or treatments or surgeries, then that's obviously not a sustainable situation. So to go along with the expansion in testing, we obviously have to have, to go with that, some reasonable decisions about how do we act on these data, and at what point is it worth it to society to intervene on a population level. Of course, individuals can make that choice if they're going to pay for it out of pocket, but that's not the case for most people.
We got onto this from, I'm going to come back, about added value of genetic information compared with other interventions possibly. I want to come back and ask about in plant sciences. Are there obvious examples of added value that you can give now from the genome analysis that has been done?
Yeah, I mean, plants is a very different. It's been very interesting listening to all the discussion about the human genome, which is, it's a very good product. In the case of plants, there are at the moment about 2 genomes that approach the quality of the human genome. One of them, Arabidopsis, has a genome of about 110 megabases, so it's relatively small, but it's a plant that is very well studied genetically. And there are traits that are studied in that plant where, for example, there's an understanding of some of the features that would increase yield. There's a lot of studies in photosynthesis. and identification of genes involved in those sorts of processes are now starting to be looked at in plants where we're interested in improving crop yields. So there is translation from the model studies into the plants, as I say, we're actually caring about.
Would this actually be just by simply saying selecting plants according to their natural genome? This is not GM, so this is something which Europe can plant and grow?
Well, yes, and that's something that we desperately need, because if GM really isn't going to be allowed, then we have to have another way of getting, of selecting what we really do need. So, yeah, I think that the estimate is that if you use GM, you reduce the time that it takes to actually sort of introduce or apply genetics through to full breeding and getting advantage out of the other end. I think with GM, you reduce it from something like 30 years down to sort of 10 or 15. But with the selective breeding, the testing in between, using testing as a sort of surrogate for real GM, you can reduce down from about 30 to 20, I think, 20 years, that is, in terms of getting these products through. So it's a long-term process.
There is, yeah.
What are the specific challenges in plant genome sequencing? Polyploidy, for example, is common in plants.
Polyploidy is one of the challenges, a large amount of heterozygosity, and sort of outbreeding being the norm. I mean, humans are outbred, but our genomes seem to be pretty well behaved, really. And the other big challenge is the proportion of highly repetitive sequences in the genome. Some of the genomes are very large, and I mean, barley genome is 5.3 gigabases, so nearly twice the size of the human genome. It's diploid, but it's got about, I think, 70% repeat content in there.
How long does it take to sequence a particular plant gene?
It depends what you call sequence.
Yeah.
You know, you can pop it through one of these Illumina machines, and it takes 10 days in the same way that the human genome takes. In terms of something that's usable, it's and useful, takes longer.
You have the big advantage of plants, of course, is all the things you can do in phenotyping. You can infect it with your favorite disease and see what it does, and they don't tend to encourage that in humans. And grow it. If you've got a hypothesis about a gene, you can grow the thing up very quickly and see what happens. Also, for understanding function, that gives you lots of big advantages that we don't have in humans.
I wonder if I could ask a question which goes back to a misspent youth in theoretical physics, where about 30 years ago I got into parallel computing, and we had one of the first parallel supercomputers with 2 megabytes of data, not just per processor. This was the total, right? So what I haven't got a feel for actually is what really is the computing challenge. I suspect there are many answers to that, because clearly and you're generating data which looked as if it was increasing faster than exponentially, and if it's just exponential, then the exponent is certainly bigger than Moore's law, than the computational power available to you. So but what actually is it easy to say what the computational challenge is and the extent to which, and of all kinds of related things, which is what are the tools and algorithms that you're actually looking for to help you to meet the challenge which raw power alone isn't going to do, right? Is that a personal interest? Has anyone got anything to enlighten me?
I mean, I can have a go. The kind of challenges everywhere. It's hard to split them out. So most of the challenges come down to 2 things. One is the sheer volume of data, and the second is the sort of high dimensionality of the problem that we're trying to solve, which is what are the set of variants in this individual's or set of individuals' genomes. So the approach that is generally taken at the moment is a very modular approach, which is very sensible because you can break the flow down into a whole bunch of things which you can parallelize and which you can put into a pipeline in a sensible way. And people are coping, and actually even reasonably small operations are coping remarkably well. You don't need Hinkston to do thousands to actually sequence a couple of thousand genomes. You don't need it. But having said that, we know that there are many areas where we're not doing very well because we're not integrating all the information that we know about where variation exists already. We're not integrating de novo assembly and mapping. We're not going back to the mappings and updating them with what we've learned. We're sort of doing very piecemeal analysis, which gets fed down a single thing and it doesn't percolate backwards in a very helpful way. So I think where I'd say is the message is we're coping, and we're coping remarkably well, but there's a lot of opportunity at every stage, essentially, to do things better.
Yeah. So a slightly different thing would be the storage of the data. So a lot of the times, or what it seems to me, is that people say it's, rather than storing, say, the images that come from the Illumina machine, it's cheaper just to bin them and to redo the experiment again rather than to purchase the storage that's necessary to actually store that. And is that going to be acceptable in clinical trials where you might just bin the raw data essentially? Is that going to be a limiting step when you try and apply this in a more applied clinical setting, perhaps?
It's like it's in clinical trials. There's good precedent for this in the current whole genome technologies. So we don't keep, for example, we don't necessarily keep the image files that are produced from whole genome genotyping, and that's acceptable. So we don't have to go back to those source data currently. So I would say that whatever happens in resequencing, there's precedent for it.
Yeah, you perhaps purposely sort of collapse the process a little bit further than I would do. I think absolutely the images are so memory hungry that we're not going to keep them. In fact, with the latest machines, we don't get them off the machine. You can choose to, but you slow your run down if you do. But it doesn't actually automatically mean if you don't store the images, you have to do the genome again, because the point is more to take the full complexity and richness of the raw data, but in contrast to being able to store it, as in the trace archive in the past, when you could go back, you have to accept a level of immediate interpretation which is validated, which has a level of quality attached to it. But then you store that, which takes a lot less information. In most cases, and I think this relates to another challenge which I'll just mention in a moment, in most cases it's a gradual process to have everybody happy with what metrics, what values are really stored, to have confidence in that information. And it's also important at the end of the day that no single result is a life-changing decision. You've got to have redundancy, consensus, whatever it is. So you don't, based on one snippet, one read, you actually have a bundle of reads that say that's why you're taking that decision. So you take away some of the risk of throwing out raw data and still interpreting it. The other challenge, though, which is what I think the whole community is grappling with, and the man sitting next to you is one of the forerunners in this whole area, is the technology is changing so rapidly that the rules built and the metrics established for the quality have to adapt or faced a year later with a changing chemistry. And I'd like to think that will stabilize in time, but nevertheless it puts a harder challenge on the metrics actually being future-proofed against such changes. You want really, really good fundamental values that will capture that quality in perpetuity. And the genome is in perpetuity. Ultimately, the data is digital, unlike any other biological information. It does have a right and a wrong. And one is simply trying to capture the nearest approximation to that. And therefore, you can sequence a genome in 2010, and the data will still be largely right in 2050. There will be improvements, and the degree to which you can track those improvements is the real challenge here, rather than looking to either store the raw data or redo it. Perhaps the way to establish whether it's still valuable is to redo it in a subset and then see if it still looked right 50 years ago.
So just a sort of flippant comment, if that it's now more expensive to store the images than just redo the data, hopefully sequencing costs will get so cheap that it won't be worth storing the processed data, and you would just resequence every time you wanted the data. That's my wish.
It's the counter view. Well, can I say, I mean, I bet you never thought you'd come here to hear this. but there's a fascinating parallel here between the analysis problems that they have on the Large Hadron Collider in elementary particle physics, where now that you've made it clear, in a sense they have to adopt exactly the same trick now, because that machine produces even now thousands of millions of collisions per second, and these collisions produce megabytes or goodness knows how much more of a data. I mean, they have detectors which are bigger than this room, and these detectors can't cope with that amount, and they therefore have electronic triggers which throw away all but 1 in 1,000, 10,000 of the events on the basis of this doesn't contain interesting physics. So there is a cut made in the data that they actually store, which seems to me like a parallel in the cut that you're making in the data that you extract from an image, but you've got to be very careful that you're not throwing a few babies out with the bathwater of the stuff that is actually not being stored. And there are fascinating questions there of prior knowledge that you're using to retain data from images or whatever. But quite an interesting parallel with the Large Hadron Collider. Rather glad to have made that. Bringing us back down to Earth. A few more minutes to go.
But they'd known this 15 years ago, and they built a network and the storage system which is specific for them.
Yes, I know. I chaired the UK
Not the case
eScience.
that-
I chaired the UK eScience program, which attempted to bring coherence across all of the subjects. And the particle physicists were in the lead because it was simple, homogeneous. They knew what they were looking for. And the biosciences are there now too. It's okay.
Not all of them.
Not all of them.
No, there are some where diversity is really what you want to still continue to capture. I think some of the population studies, the bacteria, viral, etc. You want to capture all of that data and use it to map change.
Any other questions that people? Yes, please.
Well, this maybe a silly question, but the question of the first presentation was when or if you want to sequence yourself. I'd like to ask, how often are we supposed to sequence ourselves? Because one time maybe not enough. Another question is, we have human metagenome, which is another, probably one potential interest for pharmacogenetics. Right? So do you know if this is an issue? Well,
As far as methylation in pharmacogenetics, there have been very few studies in that area yet, and I think there have been a few cell line type studies that have shown that methylation patterns can have a huge impact on some drug metabolism, certainly through expression of metabolizing enzymes. But I can't think of any studies that have been published in humans. But I'm sure that's to come. Just like everything else, as soon as we can measure it, we study it, and we're just getting to the point now where we can start to measure it.
Yeah, so the question you're asking is, which self should we sequence?
Yeah.
Because we have many selves.
Yeah. Also,
we have different tissues.
We change with time.
Yeah,
so.
absolutely. I mean, there are gross changes. Men tend to throw away Y chromosomes as they get older in somatic tissues.
So I think the story is that the problems and the challenges are just opening up. The more you know, the more you realize you don't know, which is a great way. Maybe I think if we call this part of the proceedings to an end, there will be an opportunity for all of you to mingle, and the proceedings will become more informal, and I hope will become equally valuable. Just before you all pack up, various things from me. First is, in your packs, please, there is a sheet, I think there's a green sheet, which is the questionnaire. Could you just take 30 seconds or whatever it is to fill that in and leave it on the desk outside before you go? Be really grateful. Second from me is just really by way of thanks, obviously, to the speakers and panelists and Chris for chairing. I want to thank Jill particularly, as I said before, and Robert Lees from the Knowledge Transfer Network that we run these workshops in collaboration with. I want to thank them very much for the contribution to the organisation. I should say that we are supported by various research councils, in particular, apart from our main core funding from EPSRC, we've got specific funding from PBSRC and from MRC for this programme generally. And for this particular workshop, we use what's called the Higher Education Innovation Fund to support this. But the final person that we really ought to thank is Naomi Clark. Naomi's not here. She did all the legwork for ensuring that you got here and the speakers got here and the things work. So I think we should give a round of applause for everybody. Thank you for participating as well.